Device Talks. Welcome back to Device Talks Tuesdays. We've got a great episode for you. Very popular one. Thank you to everyone who's registered and joined to hear biocompatibility testing. What you need to know. Brought to you by our friends at Nelson Labs, Cetera Health Company. A few housekeeping things before we begin. There's a PowerPoint that we'll be going through. It is available to you right now in the resource widget, so go find that. For those who are listening live, thank you very much. We love having you here at 4 o'clock Eastern Time. For those who had to fit us into a different time slot, we thank you for doing that. You can still ask questions. I'll get into other Q&A things in a minute, but if you're watching this on demand, feel free to ask questions. Type them into the Q&A chat box, and those will go directly to the fine folks at Nelson Labs. As you're watching live, feel free to type in your questions as they come to mind. We will likely address them all at the end of the discussion, but we love to have them all queued up so we know how many we're dealing with. So again, don't wait until the end to ask your questions. Type them in right away so we know immediately what your questions are. Finally, per my earlier on-demand comment, this is available on demand. So if you're watching this live, again, thank you. And if you're realizing that a colleague, friend, a family member, or anyone would benefit from hearing this discussion, watching this discussion, and asking their own questions, let them know it's not too late. They can go to devicetalks.com. Sometime tomorrow afternoon, we'll have it. They can register anytime. We'll have it available to them to watch at their convenience. So feel free to spread the word for us. We'd be very appreciative. So real quick, Device Talks has podcasts, lots of podcasts. Go to devicetalks.com to register for that. I'm sorry, to listen to those. You can also find those on any major podcast player. You will be able to register for Device Talks West in a couple of weeks. We'll have more information on that, and we'll have information about that on devicetalks.com. Without any further delay, let's get our presentation started. Again, brought to you by Nelson Labs, a Cetera Health Company. We've got a great panel for you today. Lucas Ma, who is a biocompatibility expert at Nelson Labs. Dr. Katrina Hurst, a biocompatibility expert at Nelson Labs. I have to slow down. Don't talk so fast, Tom. And Audrey Turley, who is Director of Expert Advisory Services at Nelson Labs. So you can find their bios in the resource widget as well. And with that, I'm handing the microphone over to Katrina Hurst. Katrina, thanks for being part of Device Talks, and take it away.
All right, thank you. Good afternoon, everyone. Thank you for joining us for our webinar today, Biocompatibility Testing and What You Need to Know. So I'm Dr. Katrina Hurst. You can also call me Katie. We also have Lucas.
Hello.
And Audrey joining.
Hello.
So today, our webinar is more of a discussion panel. So all three of us will be participating throughout. And the first thing I wanted to share before we really dive in are the standards that we will be referencing and using throughout the presentation. So those are there for your reference. And with that, I'll actually turn over to Lucas to start us off with our discussion.
Great. Thanks, Katie. So I'll just be going over just a bunch of considerations that we have to talk about for sample submission. I know a lot of work is needed, a lot of legwork is done in order to determine what kind of testing has to be done, if it even has to be done. And a lot of times when we get to the point where we say, yes, testing is needed, a lot of people just say, okay, the hard part's done, and I wish that was the case. When it comes to sample submission, there are actually a lot of considerations that we have to take into account for. And if you're less familiar with the process, it can actually be very daunting and confusing. Testing can be kind of this nebulous entity or black box where you submit your samples to the lab, and the lab just takes it from there. They can figure out all the test prep, the extraction parameters, everything. But there's actually a lot of input we need from the sponsor as well. And so a common question I get is, do we really need to perform testing when the material supplier has stated that it's a medical grade, it adheres to 10993, or they even did their own testing. And these are perfectly valid questions, but whenever I look at these tests, if they're available, they do testing on the raw material itself. And in most cases, this is insufficient. And the reason for that is because 10993 actually states, they state that you should be performing testing on the final finished device or something as close to the clinical device as possible. And the reason for this is because when you're testing a full device, you're not only accounting for the materials, you're accounting for everything that the material may be exposed to or undergoes. And something that a lot of medical devices undergo are the sterilization modalities. If it's EO tested, if it's gamma tested, they should be undergoing those sterilization parameters before testing, and they should be tested sterile. I have seen devices where they are actually approved for multiple. So not only can they go EO, they can also go gamma, and both or have one go EO and gamma. And if that's the case, then they should be seeing both of those sterilization modalities. And what this does is, if you're putting something like that on test, you're essentially accounting for all of the EO, all of the gamma. So you're also just making sure that the material is stable and there are no chemical alterations that may increase patient risk. And when it comes to the physical component of the device, we also have to consider the surface area or the weight. When it comes to extraction based methods, there's actually a minimum amount of test media we have to make sure that we have. And this is so that we can ensure that we have enough for dosing the animals or the plates. So that's something that we need the sponsor to provide us as well. And the size of the sample the physical size of the sample is also something that we need to consider. And for reasons for this is because while the test vessels that we use, even though they are validated, there's a physical limitation to them. These are just jars. And so if you have a rather large medical device, we have to then consider how much manipulation or how much disassembling of the sample would we need to perform in order to properly test it.
Yeah, because I think these are really good points that you've brought up. And I just want to talk for a minute about the sample size that we talk about and what's needed. So when you receive a quote from Nelson Labs, it says typically for biocompatibility, it'll have like a surface area that's needed. And I think that could throw some people off, but that's how we determine the amount of volume that we need for the test, which helps us make sure we have enough fluid to apply into the test system. So if you see that test quote and you're like, well, it says surface area, but I really want to know how many samples I have to send in, work with your engineer and figure out how much surface area you have, and that will tell you how many samples you need to send in. And of course, you can always ask us more questions, but that's how we tend to communicate the number of samples, because it's more about surface area than it is about an exact number. You know, like if you're talking about a lot sampling, we're not doing that. We just want to get a certain amount of material in ratio to our extraction media.
Right. And
just to add to that. Oh, sorry.
Go ahead, JD.
To kind of add to that, you're the expert on your device. We don't know how much surface area your device has. And so that's why we need you to give us that information so that we can perform the test properly. We know the testing and you know your device, and together we can make it happen.
Yep, that's right, Katie. Thank you. So moving on to the next slide, I listed some examples of some bad examples and some good examples that I've actually seen in my biocompatibility career. So starting with the bad examples, these are ones that don't adhere to 10993-1, and they could be just improper traceability, or just at the top of the list is my favorite, where an engineer gave me a cup of grease and just asked me to test it. So if you were to take the sample and compare it to a checklist as to all the things that we have to consider, a cup of grease would not check off any of those. It's not a final finished device. It was just the raw material. It hadn't been sterilized. I would have really no idea how to sterilize it properly because it would just leak through a Tyvek pouch if I tried putting it in. And so, and needless to say, his finished device wasn't a cup of grease, but it was just a small portion of his entire device. So putting that on test, while we could have figured out how, it wouldn't have given us any kind of information that would be relevant to the finished device. Another example I wanted to bring up would probably be my fourth bullet point, where everything seemed to be okay. These devices were fully assembled and provided to me. However, they were missing any kind of traceability documentation, meaning there was no lot number, there was no serial number. And this can be very important because when we're logging in the samples, we actually record any unique serial numbers as well. And this helps us show which exact devices were actually put on what kind of tests. And so when I reached out to the engineer to ask for this proper traceability documentation, that's when I found out that they had not built it on the line or in the clean room, but rather they had just gone to their desk and assembled it there. So it didn't have any of the interactions it would with a standard manufacturing machinery. It wasn't done in a clean room, so potential particulates or contaminants could have been present. And so it's definitely a less than ideal sample to provide. And I just want to stress that it is very important to be able to represent the final manufacturing processes, because contamination can be a legitimate concern. I remember getting some samples with an orange dust on it, and before testing that, we needed to identify what that orange dust could have been. So after submitting to the chemistry department, and having them undergo some analysis, they determined that it was actually Cheeto dust. So far from a finished device that would go into a patient, definitely don't want any kind of Cheeto dust or any kind of foreign matter being present in your testing. That would definitely complicate things, and testing is very time consuming and pricey. So we hope to get it right the first time. And on the other side of the spectrum, we have the good examples, which are just very straightforward. Final device is just in their clinical packaging. It was sterilized, very easy just for the lab to get a hold of and put on test. Going back to more complex, large devices, if for any reason your devices need to be disassembled prior to testing, we do ask to give us any kind of proprietary or special devices that we may need in order to make that testing possible. There was an instance where there were about 500 cartridges provided to us, or to me, and I wasn't testing the cartridge, but the staples inside the cartridge. And so the only way for the lab to get those staples out of the cartridge was for the engineer to give us the actual handle that would fire the staples from the cartridge. So, but rather than just giving us one and having one technician fire 500 times, they gave us 10. So it was a huge difference, and it really helped move the testing along as well.
Yeah, what I really like about that example, Lucas, is that we were, by testing these staples that were fired from the cartridge with the device that fires them in the clinical setting, we're able to test all of the things that can be impacted by that. And so not only are we looking at the materials and the processing of the staple, but also the packaging and how that might impact the biocompatibility, how the triggering or whatever's causing the staples to eject out of the cartridge. All of that was able to be included in the testing. We can say we've accounted for every part of the manufacturing processes, as also the packaging, and even when we're putting the device into the patient, we're accounting for all of that by doing testing on those types of samples. And so that's why we really emphasize this final finished device, because we need to be able to address all of that in biocompatibility. Those are all concerns that need to be addressed.
Right. Yeah, Katie, and that's a very good point you bring up too. The reason why we actually had to perform that testing was because the initial testing that we did on the staples, they were just manufactured off the line and then provided to the lab for testing. And a reviewer saw that and said that, and they just wanted us to redo all of the testing because it didn't have any of those interactions with the cartridge itself, even though the cartridge was a solid device.
One thing I wanted to bring up, because I love talking about these examples of samples that we receive, is that we can honestly test any of these. It's not a matter of can we test them. It's that if we test them, are we actually going to answer the question that is trying to be solved right now? And so like with the cup of grease, right? We're not, like you said, we're not putting that on the patient. It's just going to be a small amount on the device. And we want to know if that impacts the device's biocompatibility. So just testing the grease, I can already tell you it's going to fail because grease in and of itself is not meant to be injected or applied in these test systems. So we can do different ways to answer your question. So I would say don't hesitate if you want to investigate certain things to reach out to the lab and ask like, hey, is this the most appropriate way to answer these questions we have? But what Lucas is really talking about is when you have a device you are submitting to an agency, you know, we have to have a final finished device that goes through all of the processes in its final finished form. So just good things to keep in mind. We love to do experiments and we can do that, you know, where we're just answering these cool questions, trying to help you troubleshoot a specific scenario. But for a final finished device, we want it as clean as possible.
Right. Yep. That's a very good point. So for preparing samples, the photo that I've provided on the left are actual devices that the lab has at Nelson Labs. So you can see it's nothing too advanced. It's very basic things that we can use to measure and cut. So hence why if there's a very complex device that needs some unscrewing or anything like that, please make sure that we have the correct tools to do so. I remember having to use a hacksaw to just saw through an entire shaft of a device that was multiple layers of stainless steel. Very difficult, definitely would have preferred an easier alternative, but the hacksaw was really the best thing. So just going off of the idea of cutting actual samples, you want to be mindful of how we're actually prepping these as well. If you were to give the lab a sample with no explicit instructions or anything, they will prepare it at their discretion. So it may vary from sample to sample, but it's all to ensure that we are fitting them into these test vessels and having them properly extracted. So areas that I would recommend against cutting would be any kind of cured area. And the reason is because the material that's underneath the cured coating or the coating or that layer would normally never touch the patient or any bodily fluids. And there's actually a specific example where I had a device that kept failing cytotoxicity tests with floors. And despite multiple retests, we couldn't figure out why until the engineer disclosed that there was actually a zinc component that was normally coated with nickel Teflon. And we found out that that specific component was being cut every single time, thereby exposing the zinc. And so the next time when prepping, we gave explicit instructions not to cut that component, and it passed with a 0. So really, these things really do matter. And if there are any kind of doubts or questions you have, I definitely recommend reaching out to the lab with any of your questions. I'm sure they've seen much stranger things than I have and would be happy to help you before you submit these samples. Katie or Audrey, did you have anything to add to?
Yeah, I was going to say that, you know, we always ask for surface area because if you'll look on this picture, these are actual tools I took a picture of for a presentation, but we measure surface area with the ruler and a micrometer. So pretty archaic instruments compared to what the technology that's available today. And so I just want to urge you, when you're asked for surface area, please send it if you can. We're happy to calculate it if that's what is needed. But we do prefer to have that be done, through your engineers or with the software that you have available in your manufacturing facilities.
Great, thank you.
Yeah, I had one other example there where it was a stainless steel device that was failing cytotoxicity because it was being cut. And when the stainless steels are passivated, it creates that layer on the outside. But once you cut it, you're exposing stainless steel that hasn't been passivated. And so that was part of their issue, something you wouldn't expect, but that's something that happens. So definitely something to be aware of.
Yep. Thanks, Katie. And so with that, I will pass the presentation over to Katie, who will be discussing certain things that you may encounter during the actual testing.
Thanks, Lucas. Yeah, so let's say you planned it, you filled out your sample submission form, and you sent in your test articles, final finished, everything should go smoothly, right? Unfortunately, that's not always the case. Sometimes we still have some questions in the results or how the testing was performed. So then what do we do now? So the first thing I wanted to do, or what I want to cover today, are some of the most common issues that we see as biocompatibility experts when we're consulting sponsors. The first one we see in testing that gets a lot of questions from regulatory bodies are if there are particulates found in your extracts after performing the extraction. Another common thing is some discoloration that can happen in multiple samples or only certain samples. The third one are if incorrect testing parameters were selected, because whatever you tell the lab to use for those extraction parameters, that's what they'll use, and they might not be the right ones for your device. And then the last thing I wanted to talk about are failures. So to start us off, we have particulates and extracts. So one of the first questions I ask sponsors or the lab when we're investigating this is, were the samples cut? And is this the potential source for those particulates? So what you need to do when you see particulates is ask yourself several questions to help identify the possible source for those particulates. So the color and the size are all things that can help you identify that source. Also, depending on the amount visible, you can tell, is this something of concern? If it's just a few black particulates and you have a black plastic component that was cut, that's pretty clear where those particulates came from and that it's not clinically relevant because your device isn't normally cut during clinical use. And so then you have that conversation in a risk assessment. In some cases, additional testing will be needed, especially if your device is blood contacting, because particulates in the blood is very dangerous or can be very dangerous. Also, if you have an implantable device, you more than likely need to pursue additional testing. One of the most common tests is FTIR testing. And what that does is it tells us what the chemical makeup of those particulates are. If you've narrowed down the potential source of the particulates, it's ideal for you to send in samples. So if you're like, it's most likely coming from this component, send us a sample of that material so that we can have it to compare against. I had a sponsor that had particulates in testing. They sent it for FTIR testing, and the highest match was wall spackling. That's not what you want to see in a medical device. So give us something to compare it to, just to make sure that we can actually bring it back to the device or not, and see where that investigation takes you. And regardless of what your investigation entails, you should document everything in a risk assessment that identifies what route you took to identify where and what the form or where those particulates are coming from, and whether it's an actual risk to the patient in clinical use.
Katie, I also want to point out that testing particulates is very important because it's definitely something that reviewers look for, but we can't perform the actual testing when we no longer have those extracts containing the particulates. And the reason why I want to point this out is it's not standard practice for the lab to retain these extracts after testing is performed. So if there's any concern that there are particulates that have to be tested, when you're first submitting these samples, you can add certain instructions just saying, please retain extracts in the event particulates are observed. And that way the lab knows that in the case of any particulates, they will retain those for either for them to be shipped back to you or for further evaluation.
Yeah, that's a good point, Lucas. And to add to that, more comments that you can add into your sample submission form would be if you don't want your product cut. So, we do get particulates even when the devices are not cut, but it's very often when we have to cut products up. And we're trying to fit them into extraction vessels, what we have in the lab and make it work. So again, Lucas's example of exposing internal components that we know would be cytotoxic or would cause adverse responses in the animals. We don't know that from the lab perspective. So if you don't want us to cut it because of those concerns, please make sure you notify us on the sample submission form as well.
Definitely. Yeah. But yeah, again, when you have those concerns, tell the lab, and so that we can address it right away. And then we can also help in addressing it in a written risk assessment. We do it all the time. So the second thing is discoloration in extracts. This is something that will be questioned. You can't just brush over it. So questions are, what are the color changes seen? Is it only seen in certain extracts? And what are those possible sources? So some of the most common color changes we see are in the MEM serum because it has a pH indicator. And so if there's something coming off your device that's shifting the pH one way or another, as you can see in this picture, whether it's more basic or acidic, the color will shift from its kind of orangey pink to like a darker purple or a yellow. And that helps us know that something's changing the pH. That's a very common one. Others are if you only see maybe there's a darkening of like the polar or non-polar solution, and that may be because there's a dye in your device that is more soluble in a polar or non-polar solution. So in those cases, you can kind of identify where it might be coming from. Now, whether or not it's a concern for patient safety will be highly dependent on how your device is used and the type of contact it has. But again, regardless of what you find and what your end conclusion is, everything should be documented in a risk assessment. Okay, so another very common issue that we run into when we're helping sponsors with FDA or other regulatory body feedback is that the regulatory body doesn't accept the parameters that were used in the testing. And again, so when you're sending in your samples, you will select your extraction ratio, you'll send in the surface area and the temperature. You're selecting these in the sample submission form. So knowing how your device should be tested and what extraction temperature time ratios should be used for your device is vital. If you tell us to do it one way, that's what we're going to do in the lab. So what I wanted to do is also give you some guidance from the standards on what extraction ratios should be used depending on your device type. So if you have a thinner device, there's the 6 centimeter squared. For thicker devices, the 3 centimeter squared. And then depending on the type of material your device is made of, there is the possibility of using weight. I do caution against this because it is generally less acceptable. What really needs to happen is you need to use the extraction parameters that are going to be the most exaggerated or conservative. So if it turns out that using the weight ratio would be a lower extraction volume, then that is considered more conservative. And then 100% I would support that approach. But generally surface area is considered more conservative.
Yeah, and Katie, and you bring up a good point with the just making sure that we're testing the worst case. And I know in a previous slide of mine, I said to exclude the non-contacting areas. And the reason for that is because when sponsors have given us samples along with the surface area, they actually accounted for all those non-contacting areas as well. And so that means that with a higher surface area, there's more test extract. And not only are you testing components that would normally never contact the patient in a clinical setting, you're actually diluting that test media with all these other extracts from those non-contacting areas. And by doing so, that essentially invalidates the testing. And there's really nothing we can do other than a retest from that point.
Yeah, exactly. So if oh, sorry, go ahead, Audrey.
Yeah, I was just going to add to that when we think about worst case scenarios, it's important that every facet of medical device examination is a different worst case scenario. So, you might have a worst case scenario for sterilization and a worst case scenario for your routine monitoring, but worst case scenario for biocomp may be something different. And so when Lucas is talking about we want to make sure we're having the appropriate concentration of leachables from the device into the solution. So that's what we're thinking of. How do we get that to be appropriate? It's better if it's too concentrated. So if we have a lot of device to less fluid than the reverse, but again, you could still get false results. So we really want to try to be as accurate and correct as possible. And we can control, we can help you determine what that is, but you just need to keep that information internally.
Yeah, exactly. So one other thing to, I guess, discuss on this is, or as far as worst case scenario, going back to that. So on my last slide, I had a sample, I'll just go, manipulation. So if your sample is manipulated, then that's the reviewer is going to feel like you're removing something from the system and it's not worst case. And so they're not going to like to see that. The most common thing we see is centrifuging the samples if there's like cloudiness or particulates, and the reviewers are not going to like that. So I really caution against that as well. So the other bit of guidance from the standards that I wanted to give is the different extraction parameters of time and temperature. So you can see there are multiple ones that are given in the standard, and they're all considered acceptable. But most regulatory bodies want to see this 50C at 72 hours. And this is because this is generally regarded as the most exaggerated or most conservative. And so, regardless if your device is limited contact, there's room potentially to discuss a shorter or a lower temperature, but you will get pushback regardless if you use anything other than this. And so if there isn't a reason like material stability for not using these times and temperatures, then I would say always stick with 50C for 72 hours. The one main exception is for cytotoxicity testing, and that is because there are many proteins in the serum for the MAM elution tests that can denature at the higher temperatures. So this is the one main test where you can use that 37C. And then if your device is limited contact, 24-hour extraction is acceptable. For long-term and prolonged contact, 72 hours extraction should be performed. So that's just some guidance I wanted to share straight from the standards for you.
Hey, Katie, I just want to add that there's another caveat to the extraction times. If people are thinking, oh, I know I've done different extraction times and temperatures, that's also with hemocompatibility. So I just wanted, we're not covering that in detail today, but it is one of those tests where we have different extractions because we're working with blood. But in general is what you're talking about. In general, it should be 50 degrees for 72 hours. So I just wanted to say that there are other caveats that the lab will say, oh, we can't do that for various reasons, and that's allowed.
Yeah, that's a really good example. And there are specific standards for the blood tests that help with that, give that guidance, that are different from the 10993-12, which is what this is from. Okay, so the last thing that I wanted to discuss are failures. I'm sure a lot of you know cytotoxicity is the most common failure, and that is due to its sensitivity. But it's not the end of the world if you fail cytotoxicity. You do need to do some investigation. You can't just say, oh, well, it'll be okay, everything else looked good. You have to do that same risk assessment. You have to address that risk and give the reasoning for why it isn't a concern during the clinical use. Just some common materials that we see fail cytotoxicity are fabrics, devices with antibiotic coatings, certain metals, and then also especially for reprocessed devices are disinfectant and detergent residuals causing those failures. So with a device that has an antibiotic coating, my biggest recommendation is that you can't necessarily just say, well, we know it's going to cause a failure, so let's just test the device without the coating. You're going to get some pushback on that because it isn't that final finished device. So in those cases, what I really recommend is you test the final finished device with the antibiotic coating, depending on the concentration and other things, it may not cause a failure. And if you test that final finished device, the regulatory bodies are going to be more content with the results. And then once you've done that, you can test that device where it's final finished as far as you can take it without the coating, just to verify that it is the antibiotic coating causing that failure. So that's just some general guidance on things like that. With reprocessed devices where you have disinfectants and detergents that are causing, accumulating. So let's say you test it on the first use device, it passes, and then you have it reprocessed 6, 10, 20 times, and now it's failing. So that is, most likely it's caused by those disinfectant or detergent residuals accumulating. And there are a lot of troubleshooting things that we can do to help, minimize that. And we have, different departments that can help with the reprocessing, validations, and so forth. In general, failures, we could talk probably the next 20 minutes about failures, but there was a webinar that Audrey and some of our other colleagues did that I've provided the website for here as well. So if you want to dive into that a little bit more, please follow that website.
Katie, I want to add here too, one thing to think about any failure in a test, but especially in cytotoxicity, these tests we're trying to take the human body and separate them into different systems that we can analyze. And so it's not necessarily if one test fails, for sure another test will. These are not predictor tests. Cytotoxicity being so sensitive where we see a lot of failures, it's just because there's no recovery methods available for the cells. So even if we see a failure in cytotoxicity, it doesn't tell us what will happen for irritation or sensitization. It just tells us that something toxic is present in the device and in the leachable extract. So we want to understand what that means to a patient and if there is a safety concern. So the investigation is appropriate to look at the materials, see where we see these common things, the common failures happening, and then to understand if we would anticipate that if we pursued some of the animal tests. We have often had failures in cyto and no other failures in anything else. We've also had passing in cyto and failures in other tests. So again, it's not a prediction test. It's an informative test that leads us to gather some more information if needed.
Okay. So from here, we just wanted to offer our advisory service email. So this email you can use to contact really any one of our departments. Like we said, we have sterilization groups, packaging groups, biocompatibility, toxicology, chemistry, and by emailing this advisory services, they'll redirect you to the most appropriate group to answer your questions.
Great.
And,
all right. Well, excellent presentation. If I could put a virtual discussion up in a refrigerator, this would be it. You guys were fantastic. Good energy, lots of information. And I'll end the suspense for everybody. Not only did our panelists do great, but our Device Talks Tuesdays audience is doing an amazing job of asking questions, so good that I can tell you we're not going to get to every single one of them. So we will try to deal with the ones that we think will hit more broadly with the audience. But once again, you'll get a follow-up email if we're not able to address it on the webinar. So we'll do our best. Quick question for Lucas. I think everyone wants to know, how did you determine it was Cheeto dust? Did you kind of do a forensic? Did you follow the steps back, or did someone actually just decide to lick the device to see what it?
That was really just out of my wheelhouse. We have a very extensive library and said, yeah, this is Cheeto dust. So
okay, all right.
We're not testing that one. So
okay, good to know that no one tasted the powder. Good. Yeah. No. This is an elite core of biocompatibility experts. They're not about to do that. All right. Question number 1 from Rob. Rob asks, do you need to age samples prior to testing? I'm not sure who to throw that to, but I'll just throw it to the group.
Yeah, so that's a really good question, especially because life cycle has been emphasized more and more recently by the regulatory bodies. In general, we don't necessarily age for every test, but we have generally recommended performing cytotoxicity on an aged device versus a non-aged to have that comparison. So that's kind of the or generally what I've seen or done in the past. I don't know if Audrey or Lucas have additional thoughts.
Yeah, in my experience, cytotox testing on an aged device, whether it's accelerated or just ambient storage aged, has been sufficient. I do want to throw in, if for any reason, or if you have a reusable or a reprocessed device, that's kind of different, because that's when it really gets a little more complicated, where the reviewer wants to see any kind of potential cleaning residual buildup or anything like that. So there's a little more legwork that has to be done for those, because then you actually need to expose those devices to a certain number of tests or a certain number of cleaning cycles to show that despite all the exposure to the any kind of cleaning detergents, it's still considered safe.
Yeah, I would also add there is a requirement for MDR that we test at end of life. So we want to look at the end of shelf life, end of processing, and that's a heavy focus for MDR. It's also in 10993 Part 1 where we talk about that as well. What we don't want to have happen is for biocomp testing to be done twice, where it's on a freshly manufactured device and an aged device. That's just a lot of burden on the animals and on manufacturers. So we want to really try to assess any possible risk from the aging or reprocessing process. So that's why we start with cytotoxicity, because it's an indicator test, and then we can go from there and run further investigations. So we would not say to age samples before all of your biocompatibility testing. And we've seen it more heavily requested from notified bodies to do some aged analysis, not necessarily with the FDA. So I would say, think about where you're submitting and really start with cytotoxicity for an indicator test to let you know what other investigations are needed.
All right, great. Next question is from David, and just to again reiterate to folks, we're not likely to get, there's no way we're going to get to every question. So if you're asking questions at this point, I don't know if we're going to get to it, but if you have a question and want someone to get back to you, just indicate in that question, I have a question about X, please call me or email me. They have all your information. Don't worry about putting that in there. So, but don't be shy with your questions, because these folks are great. Okay, question from David. What if you start device manufacturing with a machine component from a solid polymer stock, and then you switch to injection molded component using the same exact raw material and polymer? Audrey, is that something? Who wants to take that one?
I can take it if you want us
okay.
to. Yeah, so this question here is really asking, how do we assess biocompatibility when we have a change in our manufacturing process? So we've started with a machined component, we're moving to injection molding. So there are differences in those processes, and we want to understand if there's different additives or contaminants that the product can be exposed to during that, possibly oil or grease from the different types of environments where the processing will happen. And so what you're going to hear Katie and Lucas and I talk about often, we'll say it in lots of different ways, but our favorite biocompatibility answer is it depends.
It depends.
So it depends. So we want to ask some really good questions, and you'll hear us follow through that process every single time. So what type of contaminants can come into contact with the material? And do we expect the leachable profile, meaning the integrity or the matricity of that material, to be changed from machining versus the injection molding? Now, during injection molding, yes, the integrity of the project or the material will change, but at the end, when it's cooled and solidified and completed that injection molding, are we back to the same material? So those are the questions that we would typically ask.
Okay. Next question from Michael. If the medical device is a gel, how is the surface area calculated? Would it depend on application of the gel? Interesting.
Yes, this is one for you, I think. Or Katie, one with the, I was thinking of the grease.
I don't want to even get into the grease story. That's disgusting. Anyway,
yeah, so hydrogels do make some interesting testing methods. So it really kind of depends. So in some cases, you might be able to justify that the weight is more conservative, and that would be easier to measure. But yeah, it depends. But Lucas, do you have more thoughts?
Yeah, I'll also throw in when it comes to certain materials like that are non-solids, but maybe they get cured or anything like that. If that's how they're going to be used in a clinical setting, then the recommendation is to perform said curing or allow the proper time for it to solidify or anything like that, so that it could emulate those settings. As far as if, when I was handed the cup of grease, I just kind of shut that down. So I can't really tell you like how that was tested, but that's something that where weight would be something that would be more accepted.
Yeah, you're right. And I think when we talk about these gels, we also want to talk about dosage to the patient. So this is getting a little more complex than we talked about in our test today. But some of those gels are water soluble. So when we put them into our extract solvents, which are mostly water, then we get this concentrated, we get the solution rather than an extract of a solid. So we want to think about testing that a little bit differently, especially if we're going to add that then to an animal. We want to be thoughtful of dosing because we don't want to just inject a certain amount by weight without any thought to what that is, considering like the amount of liquid volume in an animal. So I would say if you have a gel, you really want to have a well-documented biological evaluation plan that talks about your rationale for how you're going to sample that product in all of the different tests. It's not just one and done, because all the tests have different considerations, different animals that we have to be considerate of. So I would say if you have a gel product and you don't have a very nicely written BEP, please email us, because we would love to help you with that, because that takes a lot of coordination with the lab.
Yeah, it really does.
Nope.
Just real quick. So I've recently worked with a couple different devices that were gel, and one we ended up doing where we extracted it, and another where it was a direct injection, and it was really based on the differences in chemistries and so forth. So it really is something where having a biocomp expert and someone really guide you through that is extremely helpful.
Next question from another Michael. If a supplier sells a quote medical grade compound, does that mean it's biocompatible?
It
I
depends.
think,
I'm just kidding.
yeah,
Played, yeah.
I think that, yeah.
So-
So another valid question.
Go ahead, Lucas.
Oh, good, okay. It's certainly a good place to start, and it could give you a good idea regarding the risks. But again, it's when it comes down to it, regulatory bodies want to see testing on the finished good or have a really, really good rationale as to why you didn't. And again, because normally when they make those claims, like it's medical device grade and things like that, any kind of testing they did were just on, say, coupons or pellets. It's just in their raw form. Sometimes they're not even sterilized before testing. And again,
Yeah.
medical is such a broad term. And with biocompatibility, there's so many different levels of, or so many different categorization levels, and where depending on what the intention of your device is, you may need to address other endpoints, and the material supplier may not have done so through their testing.
Yeah, I would say a lot of companies will run cytotoxicity testing and then claim medical grade, with that being the only test that they've run. So as a caveat, in addition to what Lucas just said, but ultimately the regulatory bodies want to see a final finished device so that we're accounting for all the manufacturing processes, sterilization, packaging with that one test or the one round of tests.
All right. Follow up to that, or an add-on, I suppose, from, say, Michael. A supplier has given me an old biocompatibility cert. How long is biocompatibility testing good for?
Depends. No, I'm just kidding.
I feel like I just need a graphic to pop up on the screen.
I know. We say that. Everybody says that in their presentations. So the answer to this question is biocompatibility testing remains valid as long as you don't change anything about your device. So it's really in what changes. As we have developed the better science and we have more refined test procedures, that doesn't mean necessarily there's now this newly established risk for the patient that's going to be exposed to a device that's been on the market for 20 years that we know is safe. But if we did biocompatibility testing 10 years ago, and we've done maybe 5 or 6 or 7 or 10 or 20 small changes that we said, oh, this doesn't impact biocompatibility, I would bet you probably have a whole new device that's not been assessed for biocompatibility. So old biocompatibility certs can certainly give you some information, but I would be more curious about what has changed between the time of testing and the product that you have now. And when I think about certificates from suppliers, often they will say that it's a one-for-one change or like-for-like change. And that may be the case from a supplier standpoint that meets their requirements, but from a biocompatibility standpoint, that is rare. So I would just ask questions. I'm not saying, it's definitely not good. I would just be more curious and get more details on what has happened to the product since it's been tested.
Great. So next question is kind of a general, when should this be part of the process kind of question. So it's from Gilad. Sometimes we as designers are looking to incorporate materials into devices. So knowing if a raw material is biocompatible is really important, and we can't necessarily wait until the full device is designed. You're saying we should test raw materials first, and then at the end of the process, test the entire device, correct?
Not necessarily.
Set us all straight. Yeah. Okay.
Yeah.
It depends. No, I know.
Yeah. So, kind of like what Audrey was mentioning earlier, is cytotoxicity is maybe a good screening when you're picking materials for different parts of your device. If it's a part that does not contact the patient, then biocompatibility is not something that needs to be addressed. So it's not necessarily something you need to do for every material in your device or every component. But you can, if you want, screen just as a reassurance that the material isn't going to pose an issue in your device. But ultimately, when you're doing your final panel or group of testing, it should be on the final finished device.
I would add to that that there are known cytotoxic agents or known irritants. And the thought would be, as engineers, that when you're investigating these materials, you're looking for performance for the device, but you're also looking for what's known about that material. And this is an assumption on my part. And if it's not included in the processes, there should be some sort of evaluation of what's known about the material in its solid state, how you will use it, or in its liquid state, whatever phase you're using it in, to be thoughtful there. So we know that, you know, you don't want to get to the end and say, oh my goodness, now we finally tested it and we're failing every single test. That has certainly happened for some devices because we are using some interesting chemicals in our medical treatment now as we find ways to administer them to patients in a very safe way. But I would say it's good to be thoughtful upfront of toxicity and then go forward and do your best. Cytotoxicity is a great screening test. Again, it's not a catch-all, but it can tell you if there are toxic leachables that would most likely cause like a systemic toxicity response that we might see later. Like we get a lot of plasticizers that come out of PVC that we know cause systemic or reproductive toxicity issues later that we don't catch in some of the other tests. I would not recommend to do animal testing on the raw materials. I would strive to do some in vitro analyses first and some good research on the materials you intend to use.
We've got about 3 minutes. We'll get in one final question. Once again, if you have a question that we have not gotten to, that will go to Nelson Labs, and they will get back to you. If you're watching on demand, of course, ask your questions, and those will go right to Nelson Labs. Or if you have a question while we're answering this final question, feel free to just pump it in there, or just say, please send me an email. I have a question, and they'll get back to you. All right. Last question is from Malvika. Is there a maximum for surface area of device? You went over the surface area to volume ratio of extraction media, which makes sense, but what about the surface area of device as a whole?
Yeah,
so
Or,
go ahead, Lucas.
Take it away. Jump ball. Good.
So there is no maximum surface area, but I would say that just keep in mind that when you're accounting for the surface area, we want to make sure we're encompassing all the parts that contact the patient, or the patient contacting parts. So the surface area is based off of all those components. Keep in mind that if there are internals, that we also need those as well, especially if those go into the patient as well. So all of those, that surface area then directly correlates to how much volume is needed. And I've dealt with very large components where a lot of media is used, and there's never been an issue with accommodating test media. So Katie, Audrey, have you ever had any issues like that?
So the main example I think of is really large devices, like beds, medical beds. Can't really put that into a container to extract. So making a coupon, so a smaller version of the patient contacting surface, and then extracting that based on that surface area of that coupon that you've made. And really, you have to have strong evidence that that coupon does represent the final finished product. The processing, sterilization, or reprocessing need to be included for that coupon. So that is, for much larger devices, that's probably the route that I would recommend. But again, it really boils down to making sure we're representing the final finish, what the patient's coming in contact with.
Final word from you, Audrey, or we cover it?
I would just say for some devices that are complex and large, this is where we would cut portions of the device together. If we can't make a coupon, we've cut them in and just added representative portions into the sample extract. So again, something really important to have documented in a biological evaluation plan so that your rationale is clear and you can be consistent throughout all of your testing with your organizations, and then also so that it's clear during your submissions.
Excellent.
Yeah,
right.
if there are
No, go ahead.
Sorry.
We'll
One
go
more
over an
thing.
extra minute for you. Go ahead, Katie.
if there are multiple components and materials that are very large, making sure that the samples, the ratio between the final finished and what you're testing, the ratio of surface area is the same, is another part to include.
Great, fantastic. So once again, we see questions from Elaine and David and Elizabeth and others. They're in there. We'll get them over to Nelson Labs, so they'll get back to you. If you're watching this on demand, of course, make sure you're asking your questions as well. They'll get back to you also. But that concludes this webinar. This has been a fantastic discussion. Thank you all three for being part of Device Talks Tuesdays.
Thank you.
Thank you.
Thank you.
Great, and thank you to our attendees for not only registering in great numbers, but also asking too many great questions for us to handle within an hour. So this has been a super fun discussion to have. Make sure you register for future Device Talks Tuesdays episodes. Take a look at the upcoming agendas, and have a great afternoon, evening, wherever you are. Enjoy the rest of your day. Thanks for joining us.
Thank you. Take care.